Okay, thank you everyone. Good afternoon, and thanks Dennis, and also thank you Fred Bahe and Mario Weiss to organize this fantastic meeting. In my presentation, I will share my recent experience in using near surface and satellite sensing to monitor land atmosphere interactions from small scale to global scales. This research was funded by the National Research Foundation in South Korea and Microsoft Research, and my lab members at my university, they contributed to this presentation. Let me start with the LED sensor. Some of you might know, but many people still don't know, so let me start with LED. LED is literally, it is light emitting diode, so it is widely used as a light source, like TV. But by using the reverse mode, we can use LED as a light detector, spectrally selected light detector, like spectroradiometer. So this is, this is red, this is the infrared. It has its own spectral response. And Dennis and his former technician, Ted Ham, and Joe, now technician, they developed this LED sensor. Here, red is red LED, and near-infrared is colorless. This is near-infrared LED. And on top of them, we attach the Teflon. So we measure the irradiance. So by locating this sensor upward and downward, we can measure hemispheric reflectance. So it is bi-hemispheric reflectance is measured. It turns out that this sensor was very useful to track the canopy phenology. It was in annual grassland in California. And this is near-infrared, and this is red, spectral reflectance. And after long summer drought, when rainfall happens, then reflectance quickly drops, and grass starts to germinate. And this signal was reflected in red and infrared, and also in NDVI. And NDVI similarity was well agreed with the seasonal variation of LAI. So now I'm using this sensor to monitor multi-layer canopy phenology in forest. So this is the deciduous broadleaf forest side, and this is ENF, so evergreen needleaf forest side. My student and myself installed the 4 LED sensors at different canopy height. So we are measuring NDVI at the ground. And here is DBF, and this is evergreen needleaf forest. And we use the 4-band LED that includes the blue, red, green, and infrared. So it is 4-band radiometer. One important thing is we are quantifying both NDVI and LAI simultaneously at different canopy height. So NDVI is its NDVI sensor, it can be measured. For LAI, I will not go into the detail, but there is a fundamental equation. So I accounted for clumping index and leaf angle and clumping at shoot level and larger than shoot level. And so then I can compute LAI from the LED sensor by using the blue band light penetration. This is the result. This is a seasonal variation of NDVI measured in the forest at different canopy height. For deciduous broadleaf forest, the overstory canopy is leafless. It's because this is deciduous during the winter. And the understory canopy unfolds their leaves earlier than overstory canopy. So all these 4 sensors start to see the green up for the understory canopy. So apparently the leaf out date is similar for all different canopy height. For ENF side, this is evergreen needleaf forest. The overstory canopy is very dense. LAI is 6 to 7. So the top of the sensor at the top of tower cannot see anything for this understory canopy. But by locating LD sensor beneath the overstory canopy, then we can detect the understory canopy development. This is deciduous species. Another interesting point is the slight but measurable increase in NDVI here. And we found that at this time, there was a new needle expansion. What happened? There's very bright green new needle was expanded. And this signal was well detected by this NDVI sensor. And this is the seasonal variation of LAI from the LED sensor for different canopy for over and understory canopies. In contrary to NDVI, the LAI can detect the different leaf-out dates for over and understory canopy because LAI was computed using the light attenuation for each specific canopy layer, so it can detect the different phenological metrics for multiple canopy layers. And again, for ENF site, Ebony Needle Forest, there was a new deep expansion, and it was also well detected by LAI. So there was new deep expansion, so canopy volume increased, gap fraction decreased, and LAI increased. So both NDVI and LAI showed the coherent signals. This is the phenological metrics at both sides. Here, the black is the leaf-out date, and blue is full leaf date. Those were estimated using the sigmoid function. As I said, the LED NDVI at the top of the tower, the leaf-out date detected by this sensor corresponds to understory leaf-out date. It was significantly earlier than overstory leaf-out date. But LAI, the leaf-out date determined by LAI, was well corresponded with inside observation because it used the light penetration through each canopy layer. And one interesting point is here, the full leaf date determined by LAI was significantly delayed compared to NDVI. As I speculate, the NDVI was already saturated, but LAI increased further and further. So the full leaf date determined by NDVI was earlier than LAI. Then now I asked, what modis NDVI exactly see during this green-up period? So to answer the question, I used the Lancet NDVI to overcome the scaling issues. This is the Lancet NDVI at 3 scales. This is at this red box, and this is Lancet NDVI at this single pixel of the single modis pixels, and this is 4 modis pixels. As you see, the median value didn't change much from 0.8, 0.81, and 0.8. And standard deviation was very low, 0.01, 0.01. So the standard deviation was less than 3%. So NDVI was fairly consistent. And interestingly, there was lower NDVI values here. It turns out that this was because of this ebony species. But this signal was not appeared here because there was no ebony species in this pixel. So, what does modis NDVI exactly see during this green-up period? See, this red dot is LED NDVI, and this black dot is modis NDVI, and this gray field dot is Lancet NDVI here. And overall, there was good agreement between modis NDVI and LED NDVI. Of course, there was some discrepancy here. Anyway, so leaf-out date by modis NDVI corresponded with understory leaf-out date. So modis detected leaf-out date for understory canopy, not overstory canopy. And the slight change in understory canopy LAI increased in LED NDVI from 0.4 to 0.6. So here, this is inside observations of understory leaf-out date, overstory leaf-out date. The difference was 11 days. The LAI, the overstory canopy LAI slightly increased from 0.4 to 0.8 like that, but NDVI increased further. So modis can detect this slight increase of understory canopy LAI can have substantial impact on modis NDVI signal. So now, my lab members made the sensors and spread for several flux tower sites. It was ambitious, but it turns out that there was many problems. And for example, the birds attacked and they packed this teflon. And also there was water leakage in the Amazon forest, and there was a moisture problem within the sensor in Alaska. And in the end, several sensors survived. This is NDVI data. Inside observation of NDVI from permanent wetland, Melbourne site in Canada. And this black dot is pit bulb in Kupatai in New Zealand here. And red dot is Finland is Cut Creek from Canada. And red is Woody salmon ecosystem in Tange Range, here green. And black dot is deciduous broadleaf forest at Gwangnam in Korea, this. And this yellow is evergreen needleleaf forest at Gwangnam. So this is in-site observation, not from satellite. So it is free from clouds or aerosol. It measures every day. So now, my second toy for near surface image sensor is the camera. So it is one example, the digital camera in the field. It is very cheap, $100. Just take a photo and after some image processing, then quantify the total gaps, then separate the gaps within crowns or between crowns. So by separating the gaps between within crowns or between crowns, then it is possible to quantify the clumping index. So here we use the 3 digital cameras that measured LAI every day. I visit the field monthly or bi-monthly, and I took many LAI data in large area. So by combining this temporally continuous but spatially extensive data, then I can quantify the planetary index at ecosystem scale daily. So this data shows that the different leaf-out days for different years was well captured by this data. And also there was a windstorm event and LAI quickly dropped. And this signal was well captured. This is the just single day event. So if we use the 8-day or 16-day satellite products, then such signal might be not detected. And then I asked, I have the ecosystem scale tree LAI data. And the Moody's LAI includes both tree and grass. Then why not inferring grass LAI from these 2 products? So this is grass LAI, so Moody's LAI minus camera LAI. It shows realistic seasonal pattern and also good magnitude. And this is the in-site observation of the grass leaf-out day. And this signal was also well detected. So there is some promise to synergistic use of this Moody's and this camera data. And now let me move to image processing in digital camera. So when we measure LAI using the camera, we basically measure the gap fraction. To measure gap fraction, we need some ideal histogram like this. Good contrast and minimal over or under exposure and full use of bit depth from 0 to 256. But many people have used the JPEG for this gap fraction measurement. But JPEG is 8-bit. It is in camera JPEG is under black box because the companies don't want to reveal their some specific algorithms. So it is in black box. So it is non-reproducible, and it focuses on making some good photos for human eyes. But by using the raw image has the 12 to 16-bit, and the processing raw data is very transparent and straightforward, and it is reproducible. So we compared JPEG versus raw image processing to quantify gap fraction. To do that, we need some reference data. When people have used the little for LAI or elementary equation derived LAI as a ground truth data, but in reality it has lots of uncertainty there. Even bias could be included there. So we use some apparatus. This is the aluminum plate, so perforated, so we know exact amount of the gap size. So by changing the gap size, we change the gap size by taking photos at different distance from the aluminum plate, and we also change the gap fraction by tapping this flap. So this is the result. Here is JPEG, and here is raw image. This x-axis is relative exposure value in camera, and y-axis is measured minus true gap fraction. So it is the bias. So overall, the the JPEG image shows some large bias depending on the exposure, but this raw image showed fairly consistent, in particular after gamma correction. There was apparently little bias. And this is a case of the gap fraction 0.4. And in case of gap fraction 0.06, still the raw image after gamma correction showed the less bias compared to in-camera JPEG. So we recommend using raw image rather than JPEG. So this is our recommendation: shoot raw image and convert raw to DNG format using some free software, and apply the contrast stretch of the blue channel of the image, and apply gamma correction, and save as JPEG, and do image analysis. Then we could get better gap fraction, and then we can get better LAI. I talked with Dennis about how to further develop this digital camera work. And after last AGU, I visited UC Berkeley, and here's Joe, a Dennis technician. He showed me this is a computer, and here's a camera. This hand is my student, Sungjun, and this is just $50, very cheap. This is nothing compared to fancy big instrument. And it includes CPU, and also it has the Wi-Fi module, and there is camera module. And also it can collect raw image. So we envisioned to spread many of these sensors in the forest or crop or somewhere. And then we could overcome the landscape heterogeneity issue, and we could better evaluate the satellite product. Okay, now I will jump to the global scale. Let me start with this fundamental famous equation by Montes. GPP is par times FA par times light to efficiency. There has been tons of studies on these two, light to efficiency. For example, Moody's GPP tie and PRI, fluorescence, and etc. And light to efficiency could be retrieved at 1 km resolution at global scale. But for also for FA par, there are a series of the nice products like Moody's or Cyclops or GV1 or Spark vegetation. So this FA par also can be retrieved at 1 km at global scale. But what happens in par? I know that Shunrin Yang has one paper in 2005 in JGR about the Moody's par product, but Moody's par some mapping, but there was no global product. Now people have used at most 50 to 100 km resolution of par map when applying their study to regional to continental to global scales. So there are huge scale mismatch between par and these two components. So first step to scale up the plot level to larger scale. I believe that generating high resolution forcing data, in particular solar radiation, is very important because solar radiation is the main driver of all land surface processes. And most land surface properties like LAI, albedo, LST may be acquired at 1 km globally, but most atmospheric forcing data are much coarse. So I'm now trying to generate 1 km resolution solar radiation map for beam and diffuse components separately for par and infrared. Here is one solar radiation model by Iwabuchi in 2006, and I developed the lookup table, and all input data of this lookup table derives from Moody's products, Moody's atmosphere, aerosol, water vapor, clouds, atmospheric profiles, and Moody's land products, albedo. And unfortunately, this Moody's atmospheric product is level 2, SWAT, so it is needed to project to sinusoidal projection. And it is very computer intensive. And then compute total shockwave par at sea surface temperature, sea surface, and then apply the DEM to correct topographic effects. So finally, I got the total shockwave par at land surface for diffuse and beam components. This is one example. Here, x-axis is the station data, y-axis is my model, and red is clear sky, and black is cloudy sky. So overall, it looks not that bad. So it is well possible to make a map of solar radiation under any sky conditions, including cloudy or sunny or whatever. So it is now possible. And now I use the FluxNet data to evaluate diffuse part. So this above panel is x-axis is tower and y-axis is my model. And this upper 4 figure is for shortwave. This lower 4 figure are diffuse part. So overall, the diffuse part looks fairly good, not that bad. And this morning there was one presenter who stressed to use the diffuse part because it enhanced the light efficiency. And we know from the flux analysis and from ecophysiological theory, but there has been no diffuse part map at global scale. So this is one example, 1 kilometer for global scale of the solar radiation map. It is just solar radiation. Now I'm using the cloud computing to generate 1 kilometer resolution of diffuse part, total part, and total shortwave radiation. And so in 2011, I developed one model. It is called the Breathing Earth System Simulator. So it is MODIS-derived product. It includes several modules that include atmospheric radiative transfer, like the solar radiation model, and two-leaf canopy representation, and far cloud photosynthesis model, ball-based tomato model, 10-magnetic model, and leaf energy balance model. So explicit computation of two-leaf temperatures. It is just all based on the first principles. There is no tuning, it's just principle. And now I'm working with Joe Berry to incorporate the fluorescence and C13 and carbonium sulfide into this test because my model used the FARPA model and we know the two-leaf temperatures. The model shows fairly good agreement with here x-axis is GPP from Christian Beer at 2010 science paper. And this is my model and it was fairly good. And x-axis here is ET from the Martin Jung's Nature paper in 2010. And y-axis is my model. And yeah, not that bad. And here is the x-axis is basin water balance ET. And y-axis is my model. So overall, also I didn't show here, but I also evaluated using the 33 FluxNet tau data, and it shows fairly good agreement. So we can scale up from this. We can scale up these first principles into the global scales. And this is one example of the monthly map of GPP and ET. So it is a kind of another MODIS product. So it is here's evaporation and here's GPP. But at the time it was just from 2001 through 2003. But now I'm expanding from 2000 through until now. So almost 13 years. Okay, here's my last chapter. Okay, let me introduce the cloud computing. So the democratizing global into sensing research through cloud computing. For example, Ranga Maineni and Steve Roney, all these big guys, has very strong computer resources. And, oops. Well, I was poor, yeah? Poor scientists like myself had one single PC, and no resource in supercomputer, and no skills in supercomputer or server or cluster. And very limited data storage in the local machine. Here, I think less than 1 terabyte. But we would like to do some global into sensing that I showed you. Now, how to do that? I was very lucky to work with the Microsoft research at the time. And Microsoft developed the cloud computing system. It is called the Azure system. And we attached some analysis tool to the cloud computing. And so now I'm using 1 to 256 cores in the cloud. And I use the storage 10 terabyte. But if needed, I can increase. Of course, it is not free. And the data location is in eastern United States. It is in Virginia because physically it is very close to the NASA Goddard Center. Downloading data from NASA active server, it is bottleneck in this global remote sensing study. The NASA staff might have no problem because they can access easily. But individual scientists like myself, it is very, very time consuming to download the data. After I moved to Korea, it was even harder, very slow. So by using cloud computing, I can just select data location in the world several places, and I select the Eastern United States. And the strength of the cloud computing is on demand and highly scalable. No queue. I think this is very important. Supercomputer is very fast, but in the end, there is very long queue. But this cloud computing is already just ready. Just click, then just walk. I can use this resource. And it's maintained by the Microsoft, not by myself, so I am free from the management. Weakness, not free, but relatively cheap, I believe. And I like this figure by Catherine van Ingen from Microsoft Research. Supercomputer user, small cluster owners, the rest of us here, yeah, including myself. So we need the individual scientists who would like to do some large-scale remote sensing study. We need some tool. I think the cloud computing could be one option. Now I have just finished one project with the Microsoft Research to incorporate visualization into the cloud. So now I'm doing data download, data process, and compute GPP and ET, and visualization, everything in the cloud. This is an example. This is Mexico for evaporation, and I can move using the mouse, and this upper and lower limits of the evaporation indicate this region, upper and lower limit. And this black line includes the seasonal variation of evaporation for this pixel. And I can move this pixel, this button, then I can change this map. This is August, but July, June. I can change the map. So this is not just showing. It can combine visualization and data analysis. So I think it is needed to accelerate these scientific discoveries. And I hope my first out data. Okay, this is the last slide. The Catherine van Ingen, she said that interesting quote. Science happens when petabytes, terabytes, gigabytes, and kilobytes can be matched out simply. This is kilobyte science, like the Hongyang Fang did in like 2200, and gigabyte science in adequate system, terabyte science in MODIS, and petabyte science in DAC. So the theme of this session, site measurement, scaling, modeling, and remote sensing. It includes from kilobyte to petabyte science, and we need some tool for data processing to visualization. Thank you very much.
Well, thank you, Will. You're lucky you didn't have an abacus when you came to the lab. So, any questions?
Thank you for the nice presentation. You showed the methods on how to, well, basically downscale the power at 50 to 100 kilometers down to 1 kilometer scale. But for that, you need to have, well, the DM at that resolution, which is fairly easy to get. But you should also incorporate ideas about the vegetation cover and so on. And we won't be able to have these variables at the 1 kilometer resolution. How do you think it would be possible to assume a certain distribution for all these unknowns so that we can put a confidence interval of what the optimal ET and GPP could be, but also 90 or 95% confidence interval so that the true value, knowing the unknown, should be within these bounds?
Yeah, first I thank you for your comment. And first, I didn't interpolate. The input data is 1 kilometer, and output part is 1 kilometer. So there is no interpolation, just the same scale in, same scale out. And for uncertainty, it is a very good question. And I think the strength of using this biophysical model at high resolution is to test the Jensen's inequality. The mean of the function is the same with function of the mean. So I can aggregate the input data as 10 kilometer, 1 degree, and run the model. And then I can test the Jensen's inequality. So there are some issues that could be tested. And of course, uncertainty is very important, and that is one issue that I am trying to do.
Other questions?